பென்சில் பேனெல்லாம் ஸ்டாக் ஆகும். திடீர்னு உங்கள் பென்சில் ஒரு பென்சில் தான் அது. ஆமாம், பென்சில் தான் சோஷியல் சர்வீஸ் அந்த பென்சில் அவங்க பென்சிலமா இந்த மாதிரி வரும்போது உனக்கு ஹெல்ப் பண்ணுறோன்னு தெரியுது. வரும்போது பென்சில் ரொம்ப ஆபத்துக்கு எனக்கு ஏன் மேடமே தெரியும். 5 வருஷம் கழிச்சு ஏதோ 5 வருஷம் முடிஞ்சிருச்சம்மா, எக்ஸ்டென்ஷன் இருக்குது. எனக்கு இன்னும் ஃபர்ஸ்ட் வந்து ஸ்டார்ட் பண்ண முடியும். இல்லை முடிச்சிட்டேன் மேம். மேடம் ஒரு ஃபர்ஸ்ட் சப்ஜெக்ட் முடிச்சிட்டேன். மேடம் போன வாரம் அப்படியே காமிக்க போகிறேன். மேடம் ஃபர்ஸ்ட் ஃபேமிலி ஒரு சப்ஜெக்ட் முடிச்சிட்டேன். சிசி கன்ஃபர்மேஷன் தான் வாங்கி ஒரு சப்ஜெக்ட் முடிச்சிட்டேன். மேடம் என்ன நினைச்சிட்டு உங்களுக்கே 2 எக்ஸ்டென்ஷன் ஓடிட்டு தேவையில்லாம அதுக்கப்புறம் சீனியர்ஸ் காலத்தில் கலைஞர் அப்போ போனால் மேடம் பழைய அடி phone work out
லைவ் அம்மா வந்துட்டியா வராம அம்மா
எங்க நிக்கிறீங்கன்னு பிளான் பண்ணுங்க. முகத்துக்கு ஆனால் ஆக்சுவலி இதில் வந்து நீங்கள் டார்க்காக தான் தெரியுறீங்க, இதுதான் பிரைட்டாக இருக்குது, ஆனால் நீங்கள் தெரியுறீங்க. இப்போ நான் நீங்கள் நின்று இடத்துல நினைக்கிறேன். என்ன இடத்துல நினைக்கிறேன் கரெக்டாக இருக்கான்னு பாருங்க. இதான் நீங்கள் நின்ன பொசிஷன்.
ஃபைன்.
இதோட ஸ்க்ரீனும் தெரியும்.
ஓகே. இதுனா நான் கொஞ்சம் முன்னாடி நகர்ந்தாலும் இது கரெக்டாக இருக்கும்.
அதை ஃபிக்ஸ் பண்ணிக்கிறேன்.
டன்.
ஓகே
ஓகே மேம்.
இது சார்ஜ் இருக்குமா சார்? சார்ஜ் எது போடணுமா? ஃபுல் டைம் வரைக்கும் ஃபுல் டைம் போடணும் சார்.
அப்போ அந்த கார்னர் அந்த லைனில் போட்டுச்சுன்னா சார்ஜ் ஆகுதா? போட்டாச்சு. எனக்கு வீடியோ ஒன்று விட்டுனா இந்த ஒரு இதுக்கே நாங்கள் என்ன பாடுபட்டு இருக்கோம்.
6 வருஷம் தெரிஞ்சுக்கேன்.
அவர் கமெண்ட்ஸ் வந்து ஃபாரின் எக்ஸாம் இருக்காங்க.
நீங்க உள்ள போயிருக்கீங்க.
இந்த ஸ்லைடு கேஸ் சொல்லிக்கிறேன். இந்த ஸ்லைடு கேஸ் சொல்லிக்கிறேன்.
இந்த ஸ்லைடுக்கு ஐ டோன்ட் எக்ஸ்பெக்ட் ஐ வாண்ட் டு see only for my photo.
அவர் போட்டோ பிளான் ஆகல உள்ளே நினைச்சது யாரா அடுத்த வாரம் நான் வரேன். அடுத்த வாரம்
மார்ச் மாசம் உங்களுக்கு கூப்பிடுறேன்.
ஒரு ப்ரிவ்யூக்கு தான் என்னோட ஸ்லைடை போட்டு சின்ன பாட்டில்
வேனே போடுறேன் அதெல்லாம் பாங்க போதுமா நீங்க எல்லாம் சரி பண்ண வைக்கிறேன். ஏமா அகாடமிக் கிளாஸ் தான் ஏன்
வர சொல்லி ஏன்டா
யார் குரூப்ல பண்ணலாம்
குரூப்லயே போட்டேன்
பாருக்கு டான்ஸ் ஆட போகிறான்னு கேட்குறேன். என்ன படிக்கிறீங்க? என்ன படிக்கிறீங்க? எதுக்கு வந்திருக்கீங்க?
यार चलता वक्त स्लॉट रहता you cannot go in between अपना नहीं को वीडियो पे अंगे पे बाय from यार next hours 12 o'clock वरके डिफेंस नडको so you cannot, it's an official proceeding, you cannot go in come out अला मनम दिया till 12 o'clock you have to be यार का जो स्लॉट्स हमारे ना प्लीज पे you can go to computation पैराग्राफ lab
is also there. It's a part. It was part of the 2012 onwards. I was working with Department of Chemical Engineering and mostly was working in bioscience. My research would be mix of bioengineering and chemical engineering. Research is totally interdisciplinary. There is no discipline at all. It's totally interdisciplinary. So you should identify the scope, what are the emerging areas where you can work, where industry need your research, what is the research gap. So how many of research scholars are there? 80 doing PhD. So this is very important for you.
A warm and pleasant morning to one and all gathered here. So first of all, I would like to invite the examiners of today's Viva-Voce examination of Mr. ASHWIN Raj, Dr. Ram Charan Singh and Dr. Siva-Subramaniam. And we are very honored to have you here, sir. And I just like to give the examiners of today's Viva-Voce examination. Dr. Ram Charan Singh, sir is Professor of IIT.
So here you may ask me why I have chosen this. So this particular substrate is actually a base for mushroom industry, but it can be utilized in some other industries like bioremediation, where it can be converted to biochar. The particular mushroom substrate can be converted as biochar and be utilized for any bioremediation application and other sources. But predominantly here in India, we are utilizing it for as a soil amendment and vermicomposting process. And apart from that, there are no much applications. So this particular substrate has been chosen for production of my biofuel, and also it is going to have much more lignocellulosic residues in that, and which is going to act as a much more sugar producing yield for this particular research, and also the waste valorization. So since I said it is going to be a waste disposal, I mean it is causing major disposal problem. For 1 kg of mushroom which is being produced, we have more than 5 to 6 kg of the waste being generated. So in order to solve the disposal problem and also to reduce the amount of waste being generated, I have chosen this particular spent substrate as my substrate for the research. And next one is it can also be completely utilized as a biorefinery potential, where it can be scaled up also. And next, since this particular substrate is a zero, like it is actually from a waste, and so it is of zero cost feedstock. When we are going to scale up the process, obviously we need not spend much for the substrate production. And next, why biobutanol? So we have seen about the substrate, and now I am going to tell you about why we need to go for the production of biobutanol. So as you all know, like we have different types of biofuels like biogas, biomethane, biodiesel, bioethanol and biobutanol. So as I said, like the biodiesel, biogas and all others are actually in a current scenario, but they are actually outdated. And the current trend in the research is bioethanol and biobutanol. So you might ask me a question, why biobutanol? So let me explain you why I have chosen biobutanol and not. So the major here, like I would emphasize is the the calorific value and the energy density. So actually for ethanol, the energy density is 21.2, and for biobutanol it is 29.8. So we are having a higher energy density, and also the calorific value is higher for butanol, which is 38 megajoule per kilogram, and for ethanol it is just only 26. So it is having a very higher property when compared to butanol. And also the next property is like when we are going to use it in engines, it can be used as such without any modification. But if you are going to use ethanol, we definitely require a modification in the engine, which is again going to cause more changes in the manufacturing companies also. So that is why biobutanol is also preferred for that particular purpose. And next is the high blending versatility. Since biobutanol is said to have a less moisture content, it can easily blend with the gasoline. But ethanol is said to have more hygroscopic value, so it will actually form phase separation, and it will result in certain problems, and hence biobutanol has been chosen.
And finally, regarding the environmental edge, so when you are going to use this particular biobutanol, it is actually proven that around 32 to 48% of the greenhouse gas emission has been reduced. So this particular biobutanol has been chosen to be one of the major hero for among the different biofuels. And now moving on, like for production of biofuel, definitely we are going to use the waste, but the waste cannot be used as such. So it has to undergo different pre-treatment techniques also, and that is going to be the major bottleneck of my research. So let me explain you like what is pre-treatment and how it is done. So as I said, like we are going to take the mushroom spent substrate, which is having a very tough layer around the called as the lignin. So lignin is very harsh to break, and we need to undergo different pre-treatment techniques like acid, alkali, and other pre-treatment techniques to break open the lignin so that we can expose the cellulose and hemicellulose. Only on exposing the hemicellulose and cellulose, they can be digested by the enzyme, cellulase enzyme, and that can convert it into sugar, and the sugars can be assimilated by the species for production of the biobutanol. So that is the major complication in the pre-treatment process, the breakage of lignin. So once we are breaking the lignin, it will provide the maximize the accessible surface area. And next, when we are going to modify the lignin for enhanced enzymatic digestibility, we can also increase the yield. And there are certain constraints also when we are going to perform the pre-treatment. We are going to use the pre-treatment like acid or alkali or any sort of thing at a higher temperature or at a harsh condition, definitely it will eliminate more amount of toxic inhibitors. So the toxic inhibitors has to be reduced. If the toxic inhibitors are present at a higher level, it will suppress the Clostridium ascobutylicum, which is going to produce the butanol, and it will reduce the yield. So that is one of the major issue, and we need to also minimize the sugar loss and reduce the capital and operating costs. So let me give you the gist of what are the different pre-treatment technologies. Like we have physical, thermal, chemical, and biological, and other subdivisions, and I will explain you the remaining in the further slides now. So this is the overall process of my research. It has been shown right from the scratch. So first we are taking the lignocellulosic biomass, and it is completely pre-treated to remove the lignin, and we are obtaining the holocellulose. So holocellulose is nothing but a combination of your hemicellulose and cellulose. So this holocellulose will be then treated with the help of cellulase enzyme. It undergoes enzymatic hydrolysis, and after the enzymatic hydrolysis, it will undergo the fermentation process with the help of the Clostridium ascobutylicum MT-CC11274 strain, which I have used in my research. And after that, since it undergoes ABE fermentation, that is nothing but the acetone, butanol, and ethanol fermentation. So we'll be having the three major components, but biobutanol will be the major component, and acetone and ethanol will be the byproducts. So that will be separated separately, and butanol will be finally recovered and utilized for further analysis. So now comes the major problems which I have addressed, which are my research gaps. So why I have chosen this particular research is like, majorly I need to know what are the different pre-treatment techniques that are available, and all those pre-treatment techniques has to be optimized also. In order to optimize it and to provide a better optimization condition, I have fixed this particular different parameters which I will be discussing in the further slides, and this optimization of fermentation conditions are also need to be checked, and finally whatever values we have been proved scientifically have to be judged for technical and economical viability, and that is the major three research gaps, and I have addressed these three major research gaps in my research by developing a novel sustainable hybrid pretreatment technology for the SMS and performing a techno-economic feasibility analysis in my process. So here comes the main 5 objectives of my research. So the first and foremost objective is to collect the raw materials and analyze the compositional analysis and to characterize them. And next, my objective is the optimization of pre-treatment. So as I said, I have taken different conventional pre-treatment like acid, alkali, ultrasonication, and microwave. And for those 4 conventional pre-treatment, I have done certain optimization studies, which I will explain you in the further slides. And the third one is like ANN model. So just confirming the data only with the optimization alone is not going to prove the results. So I have done the ANN optimization, ANN modeling to confirm the details of the optimization techniques which I have performed. And my fourth objective is after the confirmation of my results with the help of ANN modeling and optimization of the pre-treatment, I have designed the hybrid pre-treatment, which is one of the novelty over here in my research. And finally, after the confirmation of all the hybrid treatment studies, I have completely moved on my objective to simulation highlights, wherein everything will be confirmed with the help of techno-economic feasibility analysis. And this is the entire workflow of my research. So phase 1, I have split up my research into phase 1, phase 2, phase 3. So phase 1 will comprise of the first two objectives, as I said, like collection of the substrate and compositional analysis and characterization, followed by the pre-treatment techniques. And in the second phase, I have carried my ANN network modeling, followed by the hybrid pre-treatment designing and performing the different treatment, comparing with the conventional treatments methods. And finally is the confirming the techno-economic feasibility of my research. So now let me move on to the major part of my work, which is the first objective. So my first objective is to collect, estimate the composition and characterize the indigenous spent substrate of fluorodose phosphates. So first moving on to the substrate collection. So the substrate has been, the spent substrate bags have been collected from the mushroom industry, and they were, after the harvest of the mushroom, they were completely washed thoroughly, and several washing has been done in order to remove the debris completely. And after the debris removal, it has been used for, it has been sent for autoclaving process to remove any sort of fungal spores. Because we have, mushroom is one form of fungi, it will have more spores, and in order to kill the spores, we have undergone the autoclaving process. And after the autoclaving process, we have made it for moisture removal process at 70 degrees Celsius until it has reached the constant weight. And after it has, after the substrate has been completely dried, we have milled the substrate and sieved it and reduced to the size, uniform size of 1 millimeter, thereby increasing the surface interaction. And finally, they have stored in added containers for at room temperature for further analysis. So the composition analysis has been performed with the help of the NREL procedure, which is nothing but the National Renewable Energy Laboratory at US. They have provided the protocol for this called as the Siegel method, and this particular protocol has been utilized completely for the determining the compositional analysis. So under compositional analysis, I have calculated the total solid content, ash content, lignin, cellulose, hemicellulose content, and the water extractive and butanol extractive, and the results are produced over here. So this is the overall result of my first objective. So the SMS, that is the spent mushroom substrate, is, before the pre-treatment process, is said to have a total solid percentage of 94%, and predominantly we are concentrating mainly on the cellulose and hemicellulose because they are the actual polysaccharide which has to be converted to monosaccharides for the butanol production. So we have concentrated mainly on the cellulose and hemicellulose. And as I said, since this has, from this particular result, we have confirmed that it is having a higher polysaccharide content, that is higher sugar content of 59.11%. And next is the structural characterization. For structural characterization, I have performed the SEM analysis, FTAR, and XRD analysis. And these are the results of SEM micrograph, SEM FTAR and XRD. So SEM micrograph clearly explains that the structure is completely intact with the intact, and it is very rigid and fibrous because it has not undergone any pre-treatment studies. So the structure is typical to any one of the agricultural biomass which has actually been compared by us. And next is the FTI spectra of the SMS which I have done before the pre-treatment studies. So here the peaks has been culminated over here. So the orange color ring, the one that has been circled over here, corresponds to the cellulose peaks, and it has confirmed that the presence of cellulose, and the green color ring confirms the presence of hemicellulose, and the blue one confirms the presence of lignin. So we have the all the three components in my particular SMS which has been confirmed by the functional groups of the by the FDR results. And next, moving on to the XRD data. So XRD data is predominantly performed in order to find out the crystallinity index, whether we will be able to completely convert the biomass into to sugar base. So here the purple color ring confirms the presence of crystalline cellulose at the peak 2 theta of 22 degree, and the red color ring confirms the presence of amorphous structures of the cellulose, lignin and hemicellulose at the 2 theta value of 18 degree. So the summary of my first objective is that this spent mushroom substrate is an excellent choice because it is having much more amount of sugar present in it. As I said, like we are going to have approximately 54% of holocellulose. And next we are going to move on to the second objective. So second objective is we are going to optimize the different conventional pre-treatment method for the production of biobutanol. So as I explained in the previous slide, like we have different types of pre-treatment techniques like physical, chemical, thermal and biological. So here I would like to mention one of the important concepts, like while we are going to produce the mushroom with the help of this particular substrate. So actually mushroom itself will degrade a few amount of the lignin content, because the mushroom is able to produce the enzyme called lactase, which is one of the lignolytic enzyme, which will degrade the parent stock which is present in the mushroom bag, and it will help to produce some amount of lignin degradation. But in order to achieve more amount of sugar yield, we have processed the different pre-treatment conditions, which is the acid, alkali, microwave and ultrasonication. So you might have a question like why you have chosen only these 4 pre-treatments. It has been chosen because predominantly we are moving towards the major amount of sugar yield. So these particular 4 treatments are said to increase the sugar yield as per different literature survey which I have undergone. So next, moving on to the acid pretreatment. So for acid pretreatment, I have chosen these 4 acids: sulfuric acid, HCl, HNO3 and acetic acid. So again the question might rise, why only these 4 acids? So starting from the strongest acid to the weakest acid and the moderate acid, I have chosen these, and predominantly as per the literature survey, these are the predominantly used as acids. And sulfuric acid is said to be a stronger acid, and other acids are also stronger when compared to like acetic acid is one of the weakest acid. So apart from this, I have also optimized the concentration of the acids to 0.1, 0.5 and 1%. And again for dilute acid pretreatment, the range can be either from 0.1 to 1% only, and when you are going to go beyond 1%, it will result in cellulose loss and sugar, it will result in the sugar loss also. And therefore I have fixed the conditions from 0.1, 0.5 till 1%, and the temperature has been made to conform to 37, 80 and 120 degrees Celsius. So again based on the literature survey, I have fixed the temperature conditions, and the time is 30 minutes and 45 minutes. When the time is going to go, when it's going to be exposed for a prolonged time, again it will result in a sugar loss. So this particular 2 time intervals are chosen, and when we are going to fix a lesser time interval, so it will not actually result in any sort of conversion of the sugars. So that is why these 2 conditions have been provided. So now moving on to the results. So before moving on to the results, I would like to explain that I have shown the yield of the total reducing sugar and the butanol concentration over here. So after the acid pre-treatment I have done, the pre-treatment hydrolysate has been completely filtered, the residue has been taken and the filtrate has been discarded, because it will contain predominantly the lignin and acid soluble lignin and insoluble lignin and the fragments of lignin and certain sugars. But predominantly the residue has been taken into account, and this residue has undergone the enzymatic pre-treatment with the help of the cellulose, and the cellulose will just go and act on the pre-treated SMS, and it will completely convert all the cellulose and hemicellulose into reducing sugars. So once we have obtained the reducing sugars, we will carry it over to the fermentation process. This fermentation process will be carried out with the help of the bacteria, which is the Clostridium acetobutylicum MT-CC11274 strain, which is a completely anaerobic bacteria. So this anaerobic bacteria is going to act on the enzyme hydrolyzed biomass, and it will provide the sugars. So this results completely provides the fermentation process and the total reducing sugars. So with the help of this result, I can show that the best results were obtained for sulfuric acid at 0.5 concentration at 120 degrees Celsius and 30 minutes, yielding a total reducing sugar yield of 442 mg per gram and a butanol concentration of 8.3 gram per liter. So next, moving on to the nitric acid slide. So the same process again was followed, and here we have received the best results at 120 degrees Celsius 30 minutes, and we have received this particular concentration of butanol, and the same results for HCl. But here we have achieved the higher, optimized result at 1%. So for the previous two acids, we have actually attained the values at 0.5%, but here we have attained at 1% concentration. And next, moving on to acetic acid. Since as I said, it is going to be a weaker acid, but since for our confirmation we have carried out the for weaker acid also. And here we have obtained the best results at 1% at 120 degrees Celsius for 30 minutes. So this is the conclusion of my acid treatment. So the best results were obtained for sulfuric acid. Sulfuric acid has been crowned to be one of the best acids under acid pretreatment. And next, moving on to the alkali pretreatment. So again, as I said, survey, I have chosen these four bases: sodium hydroxide, potassium hydroxide, sodium bicarbonate, and ammonium hydroxide. And again, with the same concentration that I have been acid treatment also. And moving on to the results of the alkaline pretreatment. So the best results for the sodium hydroxide were achieved at 0.5% concentration at 120 degrees Celsius for 30 minutes, with a higher concentration of 7.98 gram per liter of butanol. And for potassium hydroxide, I have received the maximum yield at 1% for 120 degrees Celsius and 30 minutes, and butanol concentration of 6.49 gram per liter. And for sodium bicarbonate, I have received the best concentration at 0.5% for at 120 degrees Celsius for 30 minutes, and the concentration to be 7.26 gram per liter. And finally, for ammonium hydroxide, the best values were attained at 1% of ammonium hydroxide at 120 degrees Celsius for 30 minutes, and the butanol concentration was very less when compared to the other alkaline pretreatment, because it is one of the weakest phase. And so here NaOH has been crowned to be one of the best alkali with 0.5% concentration at 120 degrees Celsius at 30 minutes, with a higher butanol concentration of 7.99 gram per liter. And next, moving on to microwave pretreatment. So you might ask me why microwave. So let me explain why microwave. So microwave treatment is actually one of the fastest and effective method. Within a very short duration, you will be able to get a better results as per the literature survey. And these are the optimized conditions like 140, 180, 220. These are the parameters chosen. And the 140, 180, 220 has been chosen based on the ranges by the literature survey done. And the time has been chosen for 5, 10 and 15 minutes, because overexposure of the sample can lead to caramelization and biochar of the particular component, and less time will not be very effective. So minimum 5 minutes and maximum of 15 minutes has been provided over here. And these are the results of the culmination of the microwave pretreatment. So the best results were actually attained for 180 degrees Celsius at a very minimal time of 5 minutes. And here the butanol concentration was 6.9 gram per liter. And as I said, this was the best results obtained for microwave pretreatment. And finally, moving on to the ultrasound pretreatment. So to explain you ultrasound pretreatment, we are going to utilize the hertz. Predominantly, the hertz will be utilized as the main criteria over here, which will produce the cavitation bubbles, and it will go and target the lignin and break open and expose the cellulose and hemicellulose. And here 130 watts has been kept constant, that is the 60% of amplitude has been kept constant over here, and 10, 15, 20 cycles has been determined, has been the parameters over here, and also the hertz has been changed 10, 20 and 30 kilohertz, and the time interval has been 15, 30 and 60 minutes. So here the ultrasound pretreatment results are being projected over here. So for the 10 cycles, I have received the best fit at 20 kilohertz for 60 minutes with a concentration of 3 gram per liter. And for the 15 cycles, I have received the best output at 20 kilohertz for 30 minutes with a concentration of 5.26 gram per liter. And finally, for 20 cycles, I have received the best at 10 kilohertz for 60 minutes. So at the end of the ultrasound pretreatment, I can conclude by saying that the 15 cycles, 20 kilohertz and 30 minutes have been proven to be the best parameters, and is yielding a butanol concentration of 5.26 gram per liter. And now moving on to the conclusion of my objective 2. So here, as I have said, we have performed 4 pretreatments, and for 4 pretreatments, so these are the 4 best parameters which I have chosen based on the optimization studies I have performed. And I would also like to note that acid has been found out to produce more amount of butanol over here among alkaline, ultrasonication and microwave, but alkaline is also almost similar to the acid value. So next, moving on to the third objective, which is the confirmation studies of my optimization parameters whichever I performed in the conventional treatment with the help of the ANN, that is the artificial neural network. So you might ask me a question, why artificial neural networks? I have chosen because it is one of the robust and accurate tool which can produce higher results with higher accuracy also. And to explain you like what I have microwave and the ultrasound, whatever parameters I have given as the criteria, so those were fed as the input layers. So I have followed multi-perceptron layer model over here. So here I have chosen the input, here we will be having the input layer, output layer and the so input layer will be the parameters which we feed and the output layer will be the butanol and the total reduction sugars which we obtain. And the hidden layer will be nothing but the neurons which we are going to completely change and optimize based on the training data whichever we are going to produce. So for this processing, so before processing all the data, we have to normalize the data. Since we have attained all the data in different decimals and higher values, ANN, when we are going to feed the data, it will provide inaccurate results and results of different diversions. So in order to normalize the data, we have performed the normalizing parameter. So since I have used ANN over here, in ANN I have used tan H activator, which is one of the model over here, and based on this model I have chosen the map min-max function. So this map min-max function will have the scale ranging from -1 to 1 only. So either the value will be -1 or 1. So based on whatever data I have provided, so the values will be converted, it will be normalized either as -1 or +1 only. And I have used the optimizer called ADAM optimizer. So ADAM optimizer is nothing but adaptive moment estimation optimizer. So this ADAM optimizer is being utilized, used because it has a higher learning accuracy rate of 0.001 and it is said to provide very accurate results when compared, similar to the conventional pre-treatment whatever I performed. And the major parameters which I will be explaining you over here is the RMSE, which is the root mean square error, and the R square value. Like apart from 7 different parameters, I will be mainly concentrating on these two parameters because these two are going to judge the ANN model. So as I said, like these are the 7 predominant parameters which I will be explaining in the further results. So these are the performance metrics of my ANN study. So for the first acid pre-treatment, before moving on to this, I will slightly explain you what we are doing. We will be having plenty amount of data sets. The data sets will be normalized, optimized, and it will be fed into the ANN model. So first the ANN model will be completely trained. So once it has been trained, it has to be validated, and after the validation process, we need to test the data. So for the testing the data, we will be using different types of neuron. So we will be starting with neuron 1. So if the values are neuron 1, we will stop the model. Or if it is not going to show a best result, like based on the R square and RMSE value, we will move on to the next category as neuron 3, neuron 5. So here I have chosen neuron 1, 3, 5, 7, 9, 11. So you might ask me a question like why this particular, because as per ANN analogy and heuristic models, so we have chosen the odd numbers and with the with the skip of 1. So here for acid pre-treatment, I have obtained the R stat value, another one information like higher the R square value and lower the RMSE value is proved to be a best fit for the ANN process. So here for R square value, the best one has been attained at neuron 5. But RMSE value has been only obtained at neuron 11. So here we have not got a better result for acid pre-treatment. So next, moving on to the alkaline pre-treatment, here we have obtained the best fit of R square value and RMSE value at 11 neurons. So this has found to be a best fit, and so the model has been stopped over here and we have not processed for the other because R square value is almost similar to 1, almost 1. So we have got the best results over there, and so we have stopped the model over there. And moving on to the next microwave pre-treatment. So again here we also got the best fit at 11 neurons, and so the model has been stopped over here. And finally also for ultrasound we have got the same results with 0.995 R square value and RMSE as 0.65. And this graphical representation of total sugar yield. So as I said, like two major components, like two are the major output variables. One is the total reducing sugar yield and another one is the butyranol concentration. So these two are our major output. So these two outputs have been verified with the help of ANN analogy. And here this particular graph represents, so this is a regression line and it is going to be a non-linear pattern, and the data are completely mapping over here. So you will be able to see that behind the orange color dots, we will be having a slight blue color dots here. So it is completely overlapped. So we are able to get a better results for reducing sugars. So next, moving on to butanol. So here you will be able to see different overlap. I mean the data have not been overlapped. So why we are going to check mainly butanol is because our ultimate production is the biobutanol, and that is why biobutanol has been chosen over here as a main concept. And as we all know, like I mean previously as I explained, like for a total reducing sugar yield, the mapping or the overlapping was pakka, and hence we have moved on to this butanol concept. Here you will be able to see a difference in the data representation. For microwave and ultrasound, you can see that the regression line of the expected value and the predicted value overlaps. So those two results have been found to be one of the best, two best fits. And when coming on to the alkaline acid, so acid, acid to also have a better results. But when comparing the acid, like acid is actually deviating, and we do not have a best fit over here. So like I would like to conclude the third objective by showing the important points. So we have obtained high R-square value at 11 neurons and low RMC value at 11 neurons for 3 pre-treatment methods. And also there were certain fluctuations in MAE and the AME, RAE and percentage difference are those parameters. So for those certain fluctuations were seen, but those fluctuations were because of the overfitting. But at 11 neurons, they were achieved the optimized structure of other 11 neurons. And finally, the prediction accuracy, we have attained a very low prediction accuracy, which shows that this particular model is actually a very good model, stable and accurate also. Finally, moving on to the summary. So I can conclude by saying that this ANN has turned out to be a robust tool, and confirming the data of my second objective, and based on the second objective and third objective results, I have formulated my fourth objective, which is the novelty of my particular research, which is the hybrid pretreatment, and it has laid as the foundational blueprint for my particular research. So this is the fourth objective where I have designed the novel hybrid pretreatment. So like before moving on to hybrid pretreatment, like let me tell you like what is hybrid pretreatment. So as I said, like we have different conventional pretreatment. When you are going to utilize more than 2 different conventional treatment combined and perform it, it will be considered to be a hybrid pretreatment. So the hybrid pretreatment which I have chosen over here is alkali, microwave and ultrasonication. So you might ask me why not acid, because in objective 2 acid has actually shown a higher butanol concentration, but why not acid has been included over here. So let me explain you, because acid, when I had reduced the butanol concentration, it has actually produced the major villain, which is over the nothing but the inhibitors, that is the hydroxymethylfurfural and furfural, which is the major inhibitor which will reduce the butanol concentration, and that is why I have not chosen acid over here for this particular hybrid pretreatment. So these are the 3 combination of the conventional pretreatment to produce my hybrid pretreatment. And we have fixed the parameters, so those parameters were chosen the best choice for hybrid pretreatment also. So after the hybrid pretreatment has been done, so hybrid pretreatment has been done sequentially. So once the alkali pretreatment has been done, we have performed the microwave, and after microwave ultrasonication has been done. After all those, we will be collecting the samples and we will be performing the composition analysis. So at the end of this, these are the culminated results over here. So hybrid pretreatment. So as I said in the beginning itself, we are mainly focusing on the cellulose and hemicellulose because they are the polysaccharides which we are majorly need for the production of biobutanol, which is the major hero of our research. So here the cellulose percentage has actually increased drastically to 52%. So in the previous objective 1, I have explained that for indigenous we have obtained only 36.91% of cellulose, but now we have obtained 52%, which has tremendously increased because of several pretreatment process. And when we are comparing the cellulose with other pre-treatments, acid, alkali, ultrasonication, microwave, hybrid has been crowned to be the best results. And similarly, the lignin content has also been reduced from 17.9 in indigenous to 9.15 in hybrid pre-treatment. So we have reduced the lignin content and we have increased the cellulose content for the production of butadiene. So next, moving on to the structural characterization of my hybrid pre-treatment and comparing it with the other conventional pre-treatment, the best results. So all these 5 have shown the best disintegration of my particular sample, and we have also obtained increased porosity in the SEM micrographs. And next, moving on to the FPR to confirm the functional groups that are present. So I have performed this FPR analysis. So here the orange ring in all the spectra shows the presence of cellulose, and actually in hybrid we can visualize the broadening and stretching of the cellulose peak, and that completely explains the increase in the cellulose content also. And next, the green color ring shows the intimation of the hemicellulose. So hemicellulose have also completely increased when compared to the indigenous biomass. And finally, the blue color rings explains the lignin structure, and we will be able to visualize a decrease in the lignin structure in hybrid when compared to the other pre-treatment process. And finally, moving on to the XRD analysis. So as first explained, the major range is the 2 theta value at 22 degree will it will point out to crystalline cellulose, and at 18 degree it will project to amorphous cellulose, hemicellulose and lignin. And this particular purple color ring corresponds to the crystalline peak, and you can clearly visualize that for acid and alkali you can see a very sharp peak at 2 theta value of 22 degree, which confirms that the crystalline structure has completely increased in case of acid and alkali, because the amorphous has been completely removed because of the presence of harsh conditions. But when compared to XRD, microwave, ultrasound and hybrid pre-treatment, the peak has been reduced. And also the red color ring structure explains the value at 2 theta at 18 degree. So this is going to completely range to the amorphous structure. So this amorphous structure, you can see hump-like structure in ultrasound, hybrid and microwave, which confirms the presence of amorphous cellulose, lignin and hemicellulose. But in case of acid and alkali, you can see the hump is not present, which confirms that it has been completely washed. So this is the end results of my XRD value. So the CRI index has been completely reduced to 44%, and it has shown the best results. And moving on to the enzymatic hydrolysis. So we have completely performed all the analysis part, and now we are moving to the enzymatic hydrolysis of the hybrid pre-treatment and comparing it with the other conventional pre-treatment also. So here the green color one clearly explains that there is a very huge increase of the enzymatic hydrolysis. So once the pre-treatment has been done, the residue has been taken, it has been treated with the cellulase enzyme along with the citrate buffer and maintained at a pH of 4.8, and after 60 hours the reading has been taken, it has been performed for 60 hours at an interval of 12 hours. So every 12 hours once the data has been taken and has been analyzed. So here the highest results were obtained for hybrid, showing a sugar range of 521 mg per gram when compared to the other treatment which ranged only from 392 to 452. So here I can proudly say that hybrid treatment has been turned out to be one of the best pre-treatment when compared to the other conventional pre-treatment. So this is the laboratory scale setup which I performed for my biofuel production. It was completely done in the anaerobic conditions. Strictly because Clostridium acetabulum MTCC 11274 strain is going to be of highly strictly anaerobe, and before performing it was the waters were completely sparged with the help of nitrogen gas to remove any sort of oxygen and to provide a complete anaerobic conditions. And after performing the enzymatic hydrolysis, I performed the biofuel as shown in the setup. So once the fermentation has been initiated, it has been completely performed for 120 hours. So after 120 hours, the fermentation has been completed, and the readings have been taken every 12 hours, and the readings have been provided over here. And this particular graph clearly explains that the black dots. So the black dots represent the sugar. So here we can see a decrease in the sugar because the sugars will definitely be reduced because the clostridium acetabulum main food is nothing but the sugar. So it will completely assimilate the sugar and it will convert it into butanol, acetone and ethanol. So as I already explained, it is an ABE fermentation. So in ABE fermentation, we will have the acetone, butanol and ethanol, and butanol will be at a higher concentration when compared to acetone and ethanol. So these are the different graphs which we have produced for the treatments, and this is the final results of my objective 4. So this this table completely confirms that has overdone all the other treatments and has bagged the first place with 521.53 mg per gram of sugar, and also with a higher concentration of butanol with 9.84 gram per liter. And finally achieving the bioequivalent yield of 0.38. The theoretical value is actually 0.41 and we have achieved it as 0.38, and it has proven to be very good results when coming for the regarding the butanol production. So these are the culminations of my findings. So again, based on all the studies we have performed, hybrid has been turned out to be better results when compared to the other conventional pretreatment process. So the same has been explained over here. And finally, moving on to my objective 5. And so in objective 5, I have performed the techno-economic feasibility analysis. So why techno-economic feasibility analysis? So far we have done all the data and the research completely in wet lab. So even though we have received very good results and which is found to be scientifically sound, but still we need to confirm the techno-economic studies because it is going to completely analyze the technical factors and economic factors whether it can be moved to large scale aspect. So that is why this techno-economic feasibility analysis has been done. So this is the overall process flow sheet. So this particular objective has been completely simulated. So it is a simulation study with the help of the designer called SuperPro Designer software, and this is the entire flow sheet that explains the large scale production data. And here we have included the orange colored diagrams explain the pre-treatment conditions which we have provided, and then we have implemented the washer, fermenter, other equipments like centrifugation, distillation process and the storage units. And next, moving on to the major mass energy balance. So here we have the different components. So totally 15 components have been assessed to be the input variables for my study of the process variable. So for this particular techno-economic analysis, so I have used actually 1000 tons per batch. That will be the input amount of SMS. And here the initial value, so the difference between initial and input is nothing but the initial is like immediately before the fermentation process, whatever data which we are providing will be the initial, and the input is nothing but the components which will enter from one process to another process. So the overall input minus output value is 182, but theoretically we must actually receive the value to be 0, but it is not feasible practically, and we have received a positive value as 182. So this 182 actually says confirms that there is actually some part of component that is present present in the output which has been escaped either in form of gaseous or any other forms. So these are the list of, so as I said, since we are going to completely deal with the economic studies, so we need to assess all the operating cost of the equipments which we have implemented in the designer also. So the complete cost will be $5 million, $594 million for only the equipments alone, to design the equipments, and this is completely being generated with the help of the SuperPro software. And next, fixing the capital cost. So this is being done with the help of the capex. So capex is nothing but capital expenditure, and the next another one criteria is the opex, which is the operating expenditure. So capex and opex put together will play the major role in deciding the investment what we are going to provide, and based on that the revenue will be calculated, and the other components will be explained in the next slide. So all these studies confirm the final investment of $950 million. And this is the economic evaluation. So based on the previous data, so the total capital investment is $950 million, and the capital investment charge for this product is $192 million. And predominantly here we are mainly focusing on the revenue, so that which is being produced by the cost basis annual rate, So we'll be able to get the data with the help of the unit production cost. So the data from unit production cost divided by the like minus the operating cost divided by the production cost will provide us the profit data, and based on the profit data we will be able to find out the gross margin, return on investment and payback time period. So here I have highlighted the return on investment and payback time, which are going to be the major attractive components for the investors. So investors will completely only fetch the data based on these highlighted data. So we have achieved the data as 15.15% for return on investment. So return on investment is nothing like we have actually invested more amount of like billions in the for the plant installation. So how much returns we will be able to get will be generated, and that is 14.15. Actually, for chemical engineering related and for biological production, the data will be ranging between 10 to 20%, and we have attained as 15.15. So it is very attractive for the investors. And finally, the payback time. So payback time is 6.60 years. So payback time is nothing but the amount which we have invested must be incurred by us at one particular period. So for getting the $950 billion, it will take approximately 6.6 years. So by 6.6 years, we will be able to take the amount which we have invested. So that is what is the payback period. So coming to the simulation highlights. So as I said, like we have the variables have been loaded as input. So as I said, like 1000 tons per batch has been simulated, and for that we have obtained 146 ton batch per batch of butanol, and per year 3 batches will be run, and the selling price of butanol has been fixed based on the world market rate in the by the Super Pro designer itself to be $1.4 per kg. And the cellulose removed biomass. So you might ask me a question like, what is cellulose removed biomass? So any output, whatever the products which we receive from the after the industrial scale will not go as a waste, but it can be completely utilized, and it can we can generate a revenue from that also. So that is what is the cellulose removed biomass, which is sold at $0.3 per kg. And the mixture of acetone and ethanol, which are the major byproduct of ABV fermentation, is $0.35 per kg. And finally, we have received a profit of these particular numbers in billions dollar per year. And the final summary of my final objective. So this has been proven to be techno-economically feasible based on the data of the profit margin, the return on investment, payback time, and it has also attracted many investors. It will also attract many investors, and this has been a very good study as per the techno-economic studies. So coming to the final takeaways of my research. So I end my research findings over here. So the final takeaway which I would like to explain you is like spent mushroom substrate has been confirmed to be one of the best substrate for production of biobutanol. And optimization studies, I performed different conventional treatments, and the best conditions were also noticed. And artificial neural network has been performed, and also it has been proven to be a robust tool. And finally, the hybrid treatment, which is the novelty over here, has also produced a concentration of 9.84 gram per liter. It has been also techno-economically feasible with 15.15% of ROI and 6.60 years of payback time. So also, though we have performed many research over the end of this particular data, still we can improvise the data, which can be done with the help of metabolic engineering of the Clostridium species to increase the butanol yield, and process integration can be done along with the setup so that we can recover the butanol as and when it is being produced to reduce the toxicity level for the Clostridium species. And finally, we can also perform industrial scale, life cycle assessment for industrial validation. So these are the few references which I have used for my studies, and these are the list of publications. So the first publication has been mapped to the outcome of my hybrid treatment, and I have performed. We published the data in bioresource technology with the impact factor 0.8, and my second paper has been communicated and it is under review with the impact factor of 6.7 under biomass and bioenergy, and the third paper has been published in Biochemical and Cellular Archives. And also I have received a funded project based on this particular my investigations in the year 2021-22 with a fund of 10,000 rupees under a DNSEST project. And these are some of the list of presentations where I have presented my studies. So finally, I would like to conclude my presentation by what we save is what we have. So as I said, like spent mushroom substrate is one of the major residue which is being discarded. So once if you are going to save that particular substrate, we will be converting into butanol and we can utilize it for the production of butanol at a large scale, and we can close the carbon loop, thereby reducing the carbon footprint, and we can save our India and save our earth, and we can make the earth as a sustainable place to live on. So thank you everyone.
So thank you, Ashwin, for nice presentation.
Thank you.
So we have a couple of questions. I request sir to start. Then I will ask. Good morning to all. ASHWIN Raj made a nice presentation. Good. Also, he has done a good work. More. Actually, as I have not gone through the thesis completely, yesterday only I read something. I hope. So, any questions from the audience side?
Actually, it was an excellent presentation I ever been saying. I have a few questions. And what is the last point of Viva-Voce? Last point of Viva-Voce.
So flash point is approximately 110.
So it's based on the isomer of Viva-Voce.
Yes, sir.
And the second question is, and Clostridium astrologicum is a severe human pathogen, right?
Yes, sir. So like we have followed the required protocols and we have performed it in our laboratory, sir. Like we have biosafety cabinetry, and with the help of that we have performed with all the precautionary measures we have taken.
Thank you.
Any other questions from the audience side? Okay. Yeah, you made a nice presentation. And yeah, you have publications also for
Yes.
bio resource technology. So I have some questions. Actually, you presented the objective, right? After the introduction.
Yes, sir.
So I think, but when you presented the research gap. So same way here, after literature survey, you can provide the research gap and then your objectives is whatever. That will be better, right?
Okay.
So maybe your university knows this one, but I think you can. Okay, fine, that is fine. So you use the resource, right? SMS.
Yes, sir.
So what is the availability? Even for the example, as the local volume.
Yes, sir. Actually, like around approximately 1.6 ton of mushroom is being produced in India, and for one
All over India?
Yes, sir. All over India. This
So
is the
statistics, if you include, that will be better.
yes, sir. Like I mean, those have been interrogated, sir, with the corrected pieces.
Okay.
And 1.6 ton has been produced in India, and for every 1.6 ton of mushroom that is being produced, approximately 5 to 6 tons of spent mushroom substrate is being generated as waste by the mushroom industry.
Actually, you focused on pre-treatment.
Yes, yes.
So you said acid pre-treatment, alkali pre-treatment. Among that, alkali pre-treatment will be better.
Yes, sir.
You said, right? Because it is inhibiting acid.
Yes, yes, sir.
So have you checked the pH of your process? The other one, what is the fermentation process pH?
Yes, sir. We have actually
What is the pH?
sir, pH we have received approximately 5.8 pH, sir.
Okay, acidic, right?
Yes, sir.
That's why you can't use that acid treatment,
Yes.
it won't be better.
Yes, sir.
So, but I have not seen pH values of any plot or anything. And also the slide 30. Actually, you have done more work, but one point I want to tell you, this I think it's not wrong. So here you see, you have done 3 experiments, right?
Yes.
Showing that, so at least 4 will be better because It is just because you are connecting.
Okay.
So it won't give you any radiation. So at least some typical result you can provide with more marks.
Okay.
Okay, one result at least. So that you can get that trend.
Yeah, in between points you can
Yeah.
get a different kind of result.
Okay.
So, but this may not be the equal.
So these are all suggestions. Okay.
Okay.
And so as I said, you focus more on pre-treatment, but what about this biomethanol production?
Sir, we also concentrated on production also, sir. All the data.
In page 144, now you mentioned two phases, right? Acidogenic phase and solventogenic.
Yes, sir. Like the metabolic
You
pathway.
are not given any reactions. What are the reactions occurring and the kinetics of them?
Sir, like I will not concentrate more on the that particular part, sir, because only the metabolic pathway.
Your major process is biomethanol production.
Yes,
right?
yes.
So you can't say like that. Okay. Even though pre-treatment you focus, this is also important. So someone to be done after. No, I am giving you such at least in future you are your guide. No? You can focus on it. That's why. Okay.
You can give this problem to one employee.
Yeah,
okay.
Other PhD.
Okay, sir, definitely.
Then the 143. I think you gave the figure. The figure 4.9. I think you made the connection, right?
Yes, sir, I made that
Connection,
connection.
yeah, that is fine. And the other one, yeah, 152. You gave the figure, right?
Yes, sir.
That is for actually you have done batch production, right?
Yes,
And
sir.
you developed one process flow sheet. That is for batch or continuous?
So that is completely continuous.
Continuous.
Yes.
So what is the capacity of the biomethanol? What capacity you focus is?
Sir, 1,000 tons of SMS will be provided and
I
140.
am asking the output. Output how much you get?
Sir, 146 tons of output, sir.
Per 1,000 tons? you
Yes,
get
146
tons
yes,
of biomethanol.
yes, sir.
Then yeah, actually you said biomethanol we can use as fuel directly without mixing with.
No, sir, we have to mix it, sir.
Mix it.
It requires blending.
So then yeah, as Professor pointed out, no, you have not checked the properties of biomethanol.
Okay, sir.
Biomethanol fuel, right? So you are producing it. You characterize the characterization all you have done. That is fine. Your work also good. But what about the properties of biomethanol? All properties you have to check. Okay.
Okay,
And
sir.
I think butanol it is C4, right?
Yes, sir.
So property we are asking. Like octane number,
Yes, sir.
octane, all those things.
Okay.
If you want to mix with petrol.
So like that is actually planned to be done in the future studies.
So actually that's why I asked you acidogenic phase and solventogenic phase,
Yes.
right? So what are the reactions occurring important?
So
Because if you see biomechanical production, all will be converted into acetic acid. From
Yes.
acetic acid you are getting release. So similar to that, here what is the major
Sir,
reaction?
we get acetate and butyrate over here, and acetate and butyrate will be converted to
So
acetate.
that reactions you have to include, that is
Okay.
important. Even though known, but you have to give and what is the process kinetics, that is very much important. Okay.
Since the presentation was very lengthy, I comprised it.
Yes, actually one more thing. So you have done literature survey, fine.
Yes.
Okay. After getting these results, have you compared your results with the?
Yes, sir. Like we have almost received
You
some.
compared, but there is no table or comparison in the report. No, it is not there.
Sir, but
That's also important. See, all scholars are checking the literature. After starting the working and getting the result, they are not checking.
So it has been actually provided, sir. A table has been provided in the revised literature in the one of the
No,
pages.
I'm not asking about literature, sir. That is over.
No, I'm saying that it has been compared with the literature and included in the thesis also, sir.
Where it is?
Like I can.
I am not seeing literature. Literature only is all. Okay, so if you provide it will be better. Fine.
Okay, sir.
So, two questions only. Even actually, in your conclusion, you included the scope, right?
Yes.
Scope for
Yes, yes.
separately, if you give it will be better.
Okay.
After conclusion, scope for future work that you can give. And how you calculated return on investment?
So return on investment is actually like the total revenue minus operating cost divided by revenue into 100%.
So you have not used the IRR technique also. Then that is never technique. That is okay, fine. And what about the cost of pre-treatments? Because you are using hybrid treatment also. Normally people will use only one hybrid. So since you focused on pre-treatment, what are the costs involved individually and also hybrid? Then how it influences your final production of
So definitely I would like to say that like insurance will be bit higher when compared to the other conventional pre-treatment. But when comparing to the valorization effect, I mean the production of biomineral concentration, so the concentration is higher, and when you are going to take up to large scale study, definitely it will yield more amount of margin. So the profit percentage will be higher, and that can be tallied with the help of and also since the SMS is going to be of zero cost that will not incur more amount of substrate. So if we are going to go for a large scale process.
Okay. So my final question, can you state briefly what is the novelty of your work?
So to say briefly, like hybrid pre-treatment is the novelty over here because it has been completely designed, and I can proudly say that my work is the first work which I have done for biomineral production utilizing the SMS, and hybrid pre-treatment also has been done for this particular paper, and there are no other papers so far which has been published. So that is one of the major novelty over here.
Fine. Yeah, yes, that's good work. And he has good publications also. I think you can recommend. But Indian exam, he has to learn the questions and we can
For incorporating the cases, but at this stage you should know all these answers, because you are going to get a PhD degree in this area prior to one. One first portion is that if we compare this process with solar energy, so all our energy, conventional energy, and then economic and many other advantages are there. Solar energy, now the solar companies are the cost is less than thermal power plants. They are 2.5 rupees per kilowatt. So how you compare, where you are going to use the biomethanol and
Sir, like predominantly
where you see the scope?
Sir, mainly for transportation purpose, I focus this particular because for transportation, like though there are many solar-based vehicles also, but at long run, like for transportation purpose, we are in need of any sort of fossil fuel. So as we are having a depletion in the fossil fuel,
Solar lot of transportation problems. As I told you, EVs are there.
Yes, sir.
Solar to hydrogen. This is only for transportation. Hydrogen is storage.
Yes.
Hydrogen is not a form of energy. It's form of storage. Because solar we can't move not operate the vehicle directly. So we are using the electrolysis process, producing the hydrogen, and then using for vehicles, big vehicles, heavy vehicles like trains, and then. So when you see the biomethanol, that portion you know. I'm not asking that you are incorporating the thesis. At this stage you understand what is the use, and should explain, and you can formulate the problem based on this portion. Many processes you are using. You are saying that this is green process, but many things you are using. So one technique we use that is called LCA, life cycle assessment. With help of life cycle assessment, you can evaluate the sustainability of the process. So many times we feel that this process is sustainable. Like for one example, I can give you bioplastic and plastic. So many rooms bioplastic is not sustainable because many other processes we use. We use heat treatment, use this, that. All in all cases you emit some kind of carbon. So sustainability is another the new process you are claiming, you are proposing.
yes, sir, I have added it in the future scope, like for further studies.
So if anyone asks, you must be confident.
Okay.
And you do sustainability. You give again you give this problem to some PhD student to evaluate the sustainability, and we use some software, Gabi and many softwares are there. Open LCA is also there. You can download, you can give your data, you can calculate the sustainability. How it is sustainable development means three parameters are there. Sustainable development is very popular theme. So one is cost, another is
how sustainable, how green is the process, and then application. So most of the process we previously developed, that is only based on cost and applications, like coal. Cost it was very effective, very easy, very convenient to use, and it was easily available, and society needed that time 200 years back, society needed lot of energy. We never calibrate the sustainability. Now whatever process you are developing, the people, the organization, the policy maker, they are asking where is the sustainability, how to prove that, how to quantify that. Otherwise industries are, and government will not recommend. So this is another. Then many other agro waste are there. You are saying that SMS is free of cost, but many lot of agro waste, rice straw, wheat straw, many straw. How you compare the SMS with that?
Sir, it is almost like for SMS also we are completely replacing. SMS component is nothing but either the paddy straw or the rice straw is being used in the bag, and apart from that, other
But quantity is very huge.
Yes,
So
sir.
can we, instead of SMS, can we use? You must be confident to answer this question. No rice straw for this particular process.
Yes, sir,
SMS is
like,
best.
because lignocellulosic, the cellulosic content is actually 20 to 30% for rice straw, but for this SMS it has been increased to more than 35 to 40%. So that is how, because
So that we are going for pre-treatment also.
yes, sir, but before pre-treatment we have the mushroom. Since the mushroom is actually releasing lignolytic enzymes, that is lactase, so it already has the potential to degrade.
Again, you must be very confident, because you are going to get the PhD. Pre-treatment, what is going on mechanism, that you never explain. You are using acid base, what is going on inside? What is the science?
Sir,
Microwave.
So the major thing is like we need to rupture the lignin and break open the lignin structure to expose the
So why
hemicellulose.
some processes are doing very good, some processes are failing, unable to rupture?
Because of the recalcitrant properties, because it is mainly because of certain harsh
So
conditions.
again, it's for you.
Yes, sir.
Why ANN? ANN only use only because whenever you are choosing any optimization technique, you should first think why I am going to use that, because a lot of options are there. Some are based on a lot of data is available. Why not multi-parameter regression instead of ANN?
Sir, ANN has been chosen when compared to other techniques like either RSM or CNN, and even CNN is also available, but I have chosen ANN because of its robust technology and fast convergence and also high accuracy values when compared to other.
All are very good, but
Yes.
ANN some good things are there in ANN,
Yes.
some good things are there in as least square and other things.
Yes, sir.
So that's why I'm asking why ANN.
Sir, that is also based on the performance metrics, all these data has been calculated.
But ANN need lot of data.
Yes, sir. Like
So-
we have taken more amount of data and we have trained.
How many data?
Sir, approximately like 300 to 350 data sets.
That is again not sufficient. ANN good when you have 1 GB, 1 TB data, a lot of. This conventional techniques are also good. ANN basically when we use the conventional technique when the relationship you are fitting some, it's all converting data into some form of mathematics. So if data converting in some form of mathematics, then the conventional techniques are good. They are fitting in the exponential form, they are fitting in the algebraic form, second order. ANN, if they are not fitting, the data between the output and input, the behavior is random. In that case, basically we go for novel new optimization technique, that is ANN and genetic algorithm and metal algorithm, all are new optimization technique, and data is not following any mathematical. So that is the advantage. Then you don't have any option. Otherwise, this least square technique and many other technique, regression, and they also be very good, very accurate. Just data, you are converting data in the form of mathematics, in the form of equation, nothing else. So that we are based on the fitting. In between that data, they will get very good behavior. Beyond that data, they will not show. That is the difference between fundamental model and database model. So always you can't say that ANN is good. You told that anaerobic bacteria you are using.
Yes, sir.
But it's very difficult to maintain in the real condition. Aerobic, again, cost will be very high. Maintaining anaerobic bacteria is still. So why you didn't try for aerobic bacteria?
Sir, for usual production, the major bacteria is the Clostridium acetomucicum. Completely falls under the strictly anaerobic condition bacteria only. And
So most of the researchers recommended anaerobic for this case.
yes,
Uh,
predominantly they have used only Clostridium acetomucicum
Okay.
of different strains, and this strain has been used for the first place.
Instead of bacteria, because this is controlled process. Instead of bacteria, can we use the enzyme? Extract the enzyme from the bacteria. You have better control of the process. Bacterial process always depends on the behavior of bacteria. You can't know. Even on the same set of conditions, that is the problem with biologists. Same conditions, sometimes they give one behavior, another time it's very difficult for biologists how to decide which condition, how. Sometimes they feel that experiment fails. So what to do now? So enzymatic reactions, once you have cost is one factor,
Yes, sir.
but the better control is there. So you can explore this also.
Okay.
You include relative merits and demerits because inside knowing, because you focus on pre-treatment techniques and the novel pre-treatment.
Yes, sir.
So if you are focusing that or you are proposing that, you are emphasizing that, so you should give the more insight how these processes are taking place, how these processes are working, science of these processes. One like how acid treatment work, that may be one good PhD thesis, even a thesis. How this works, how they are working, how they are breaking. Temperature, you are saying 120 degrees Celsius.
Yes, sir.
Again, for cost point of view, it's not good maintaining high temperature.
Sir,
So suppose one example is that 37 degrees Celsius you are getting 80% efficacy and 120 you are getting 82% efficacy. So this is the engineering problem. This should always go for 37, 80 degrees. Then you have to take judgment. So you can't say that 120 always. 37, 80 degrees Celsius, 80% efficiency and 120, 82% efficiency.
but here the efficiency was very good for 20
How
and to
so we have to optimize the cost and efficacy because maintaining 120 or maintaining high pH or low pH, it's always very costly at industry. Knowledge. Actually, you never show what is the result without treating.
Sir, that was explained in the first objective. Sir.
You are showing that something happening, pre-treatment, cellulose is stable in treating. But one experiment you should include without treating. That will be the base. That will be the reference.
So that has been done in the objective.
You
Yes, sir.
already there.
Yes, sir.
The cost estimation you have done, that is just case study.
Yes.
In real, this is just a case study.
Yes.
You propose that it's just a case study and the student can investigate. some idea.
Yes, sir.
Maybe.
Yes, sir.
And so this congratulations once again, good presentation, you have done good work, good number of people, and you can also continue working in this field.
Sure.
But you should know the research gap and where we are going to use that this bioethanol.
Okay.
You have to compare, you have to compete with solar, you have to compete with wind, you have to compete with EV, you have to compete with hydrogen, not a lot of. So you should know the speciality, you should know good things. Then only you will serve. Accordingly, you have to modify, you have to make the objectives, you have to identify the research for further research.
Sure.
So once again, congratulations from my side. And again, any so one wants the doctor, he can use that title.
So I would like to invite the So thank you and Dr. Ashwin's wonderful and with us we have two eminent they have motivated like anything for the work as well as the progress they have. In this forum I am requesting you so with all the things from our department, teaching, non-teaching and supporting staff, I am heartfelt thanks to one and all who are
Give permission, like I could, can I display the acknowledgement?
Yeah, please.
So my first and foremost greatest thank and gratitude is to my supervisor, Dr. M. Prabhashankar. So more thanks to Dr. M. Prabhashankar. Next thanks to my doctoral committee member and Dr. S. Karthikeyan at TNU Coimbatore. And my next acknowledgement goes to the Indian examiner, Dr. Ramcharan sir, who is working as professor at Ph.D. Michelis Corvinus. So he is associate professor at Cyprus University. And my next thanks goes to Dr. Sivasubramanian sir, like who is acting as chemical engineering department at NIT Calicut. And a special mention to Dr. V. Arumugam sir, who is associate professor at Sastra, who has helped me in certain other research works for my study, and also to Dr. S. Gautami Ma, who has also played a major role in one of my research works. And I also thank the infrastructure provided by Citra Coimbatore, and more of my infrastructure has been provided by Banaram Institute of Technology. So for that I give my foremost gratitude for contribution to your works. And here comes the rocking people of my biotech family. So without them, like I wouldn't have been successfully able to complete my Ph.D. So they have helped me in more amount of time of requirement, and also my non-teaching staffs who have helped me in more of the laboratory works and all the providing all the research facilities. And here comes the major people, like they are the backbone of my research. At this moment, I will actually dedicate my Ph.D. to them. So I thank a special thanks to them and my in-laws and other family members. And finally, without the God Almighty, this is not possible. Standing before you to do this. So thank you to everyone for coming here and being a backbone for my works. Thank you.
Congratulations to the first candidate.
Thank you, sir. So once again, Dr. Ashwin, so thanks for completing this Harvard PhD and getting it awarded by the eminent persons in the forum. So once again, congratulations to all of you. And I also thank every one of you who is present here on your busy schedule. So thank you once again all. And I also thank finally the examiners of today's session. So thank you, sir. So thank you all.
Next class. Only you started. Thank you.
Thank you, sir.
அவங்க ஃப்ரெண்ட்ல வந்து